Hola, buenas tardes a todos. Bienvenidos de nuevo al primer simposio virtual médico sobre enfermedades autoinmunes. Y pues lo prometido es deuda. Tenemos nuevamente aquí la plática del doctor Petratos, que nos habla sobre las partes modernas de biométrica de las enfermedades autoinmunes. Les dejo aquí su video con subtítulos en español. Recuerden que si tienen preguntas las pueden hacer y nosotros se las enviaremos por correo electrónico al doctor Petratos y estas serán respondidas en el grupo de Facebook. Gracias.
Hi everyone, my name's Dr. Stephen Petratos and I'll be hosting the lecture on immunopathogenesis of multiple sclerosis. My research laboratory is at the Central Clinical School in the Department of Neuroscience, and my main focus of research is defining novel mechanisms in stopping and reversing the disease of multiple sclerosis. So the lecture objectives for today are to, one, outline how dysregulation of the immune system may manifest inflammatory demyelination in the brain, optic nerve, and spinal cord. These are the anatomical sites, the anatomical targets of how the immune system affects the demyelinating condition, which is the symptoms that patients present with as multiple sclerosis. The second learning objective for today is to distinguish between disease-modifying therapies used to treat multiple sclerosis, and right in the outset, I'll be presenting a clinical case study to demonstrate how MS patients present, how they're affected in their entire lives, and how they can be managed with our current therapeutic options that we have available to us for MS. And we'll be diving deeper to analyse and evaluate the clinical and pathological presentation of multiple sclerosis. And this is a key at being able to understand the disease and the outcomes of disease onset with regards to potential therapies following the onset of the disease from an immune mechanism point of view. But obviously, it governs how we address MS going forward, particularly since progressive multiple sclerosis is an unmet medical need, and we need to start dealing with the pathological changes that occur in the brain and optic nerve and spinal cord after the onset of the disease. So, the first lecture outcome is to outline how dysregulation of the immune system may manifest inflammatory demyelination in the brain, optic nerve and spinal cord. And to emphasize this from a fundamental understanding, I will now present a short video identifying how dysregulation occurs. In the first outset, we know that the central nervous system consists of neurons and oligodendrocytes, which are the target of the demyelinating condition from the immune system. And the oligodendrocytes myelinate the axons, which is the target. So the immune cells are dysregulated in the periphery, but they're able to expand through cytokine activation, attached to the blood-brain barrier endothelial cell surface, and then transendothelial cell migrate into the central nervous system to produce their effector outcomes. And this is by expanding the inflammatory milieu within the central nervous system compartment, activating innate cells such as microglia within the central nervous system, but also activating cells that have ended up in the central nervous system, such as T cells and B cells, to produce antibodies that target myelin specifically. And as they attach to myelin, then the monocytic cells that have invaded into the central nervous system, as well as the endogenous microglial cells, are activated through the inflammatory cascade to attach onto those antibodies targeted onto myelin and actively strip away the myelin components, producing the demyelinating effects. Of course, these phagocytes play a major role in this demyelinating aspect of disease, but they also produce bystander effects, such as producing substantial amounts of reactive oxygen species, which are deleterious to the neural architecture. And so you also see substantial amount of cell death, primarily in oligodendrocytes and also eventually neurons as the disease progresses. So, as I said in the outset of the lecture, I'll be presenting a clinical case study, and then we'll put that together at the end of this lecture, just to give you a good understanding of a classical patient that presents with MS. Now, I've given you a picture here of Nancy. So I want you to meet Nancy. I'm not going to tell you which of these two patients is Nancy. We'll define that at the end. And so I haven't put Nancy's age here, but she's a Caucasian female living in Melbourne who presented to the emergency room with symptoms of numbness and tingling on one side of her body, muscle pain and fatigue. She enjoyed various excesses in her life, including occasional drinking and smoking. And Nancy was asked in the consultation further questions by the physician, revealing an extremely stressful custody battle over her two children in the last few months. So there are substantial stresses in Nancy's life over an extended period. An MRI that was performed on Nancy when she presented was showing these white patchy lesions in her brain. So we see coalescing lesions right around the white matter components of her brain, which is resemblant of MS demyelinating lesions or sclerosing lesions. Today, Nancy has recovered from her acute episode because she's been put on a medical management plan, and she also has a persistent feeling of fatigue and residual heat intolerance during summer. So these are some of the clinical outcomes that we do see in patients with MS, and they're related to the ongoing demyelinating conditions that occur not only in the brain, but also in other areas of the central nervous system, such as the spinal cord. So let's now have a think about that. But as we do, let's start thinking and discussing a little bit about what causes MS, because we know quite clearly that MS is an autoimmune condition. So that is the immune system somehow during development has been dysregulated and has made this patient susceptible to getting an autoimmune attack against the central nervous system components. Added to that is the racial and also latitudinal variant that is occurring here. So that is to say that the patient is living in the southern hemisphere in a latitudinal component where a high prevalence of MS patients reside, and that is Melbourne. Of course, the patient is Caucasian and female, and females tend to have a higher discordance of autoimmune conditions. So most common, MS is the most common chronic neurological condition in young adults, and its prevalence is about 1 in 1,000, mean age being around about 30 years of age at onset. So think about that when we talk about Nancy's clinical case. So diagnosis occurs anywhere between 20 and 50 years of age. Women are afflicted more than men, and quite clearly, as I said, in terms of autoimmune conditions, profoundly, and MS is classed in that same category. So 2 or 3 to 1 ratio of women to men present with MS symptoms. Latitude plays a significant role, and the studies have demonstrated that Northern Hemisphere Caucasian populations are more prone to the disease, particularly in areas of greater than 40 degrees latitude on the globe. So MS is then classed as a condition where there are specific hotspots in the world, or foci in the world, where MS tends to have a higher prevalence. And this is quite clearly demonstrated considering that certain populations such as Asian, Hispanic and African populations don't necessarily present with high ratios or high incidences of MS. So ethnicity and geography play a role, and they interact in a complex manner for disease susceptibility. Having said that, if a Hispanic person actually migrates to an area where there is a hotspot, they seem to have an increased chance of presenting with MS. And so the environment plays a significant role from this perspective quite clearly. And that is also included with their descendants when they live. So second degree migrants tend to also have increased chance of manifesting with MS symptoms. The other really important aspects that need to be discussed in the environmental influences of MS is low vitamin D levels, but the predisposition to Epstein-Barr virus and cytomegalovirus infection, and patients previously infected with these viruses tend to have a higher chance of developing MS. And also smoking is another major risk factor, not so much so in the initial relapsing remitting forms of the disease, but more so in the progressive forms of the disease. Now, with regards to the MS hotspots, as I said, what we can classically see is that by far Northern Europeans such as Ireland, Scotland, particularly Scotland and Northern England have a high incidence. Also some of the Scandinavian countries, and in particular, what we can classically see is that North America has a really high prevalence of MS. In some areas in the US, we see increased numbers. In fact, the US has released recent epidemiological data, which clearly shows that about over 900,000 cases of MS have been reported in North America. And this is put in context when we see that about only, it depends on which data you believe, but anywhere between 2.3 to 2.4 million cases globally of MS have been defined. And so if there is 900,000 in the US alone, this is a considerable impact that occurs from a health perspective and a burden in North America. The other hotspot that we can classically see down here is that in Melbourne, Tasmania specifically, and New Zealand, we have a high incidence of MS, even right up to the northern sea, north and eastern part of the seaboard, including Sydney. But as we go further up the country towards the equator, we see that Northern Queensland, Northern Territory and the like do not have high prevalence of MS cases at all. So there seems to be a relationship with quite clearly with a latitudinal gradient. So what's the genetic influence of MS? So we talked about the environmental conditions. What's the genetic influence of MS? Well, this is directly related, and what we classically know after doing genome-wide association studies globally is that the HLA variants, the alleles where autoimmunity occurs, are more prevalent in MS patients. So we have identified about 100 distinct genes that confer risk susceptibility to MS. And this can account for approximately 30% of cases of MS. So autoimmunity or immunological dysregulation susceptibility genes account for the vast majority of the candidate polymorphisms. So it's single nucleotide polymorphisms that occur on the HLA alleles. And argue quite clearly for this peripheral outside-in model of disease onset. That is the immune system infiltrating into the central nervous system because it has been dysregulated and then propagating the inflammatory demyelination. And the genes specifically defined are HLA-A, HLA-DRB1, particularly. These specific variants argue for central tolerance abnormalities that regulate MS predisposition and suggest that tolerance in the thymus has been overcome, and we essentially have TH1 and TH17 responses prevalent in MS pathogenesis. So let's have a look at the immunological cascade just briefly. What we see is that any cells that have escaped, any autoimmune potential cells that have escaped the thymus at that area, or the stroma of the thymus, can enter eventually into a secondary lymphoid follicle to potentially have an expansion role when activated. So an activation cascade of an autoimmune response may actually be a preceding viral infection, or such as Epstein-Barr virus or cytomegalovirus infection, or even just a cold or a flu, to initiate the autoimmune response and expand the TH1, TH17 pro-inflammatory cells to then exit out of the lymph node and enter into the peripheral circulation, which once in the peripheral circulation can go directly to the blood-brain barrier, where they can bind to adhesion molecules using their alpha-4 beta-1 integrin receptors on their surface, attach onto those endothelial cells through the adhesion molecules, and then transendothelial cell migrate through that blood-brain barrier and produce their effector responses of inflammatory demyelination. And of course, it's these mechanisms that we've reverse translated from the human into the mouse models and define these mechanisms effectively, where we've been able to develop drugs to tackle these effector responses. One of those drugs that sequesters the potential autoimmune cells in the lymph node is fingolimod. And fingolimod is a small molecule that is a modulator of the sphingosine-1 phosphate receptor, and limits the egress of effector immune cells from the lymph node where they can expand and to enter into the circulation. However, once these cells do enter the circulation, into the vasculature, we can tackle that by using tisabri or natalizumab. Now, natalizumab or tisabri is a monoclonal antibody that actually blocks the adhesion capacity of the immune cells through the alpha-4 beta-1 integrin molecule to bind to the adhesion molecule, such as VCAM1. And by doing that, it stops the transendothelial cell migration of immune cells. The biggest problem with some of these drugs is that they have substantial side effects. And one of the major substantial side effects is potential for a neurovirulent strain of virus to produce substantial brain damage in MS patients. So tisabri was actually halted in clinical trial for some time because of that effect. And so what we can classically see, as described before, is that an altered MHC presentation through dendritic cells of autoantigens can occur through, once activated, to activate the immune cells, the CD4 positive immune cells, to then heighten the amount of production of pro-inflammatory cytokines such as interleukin 1 beta, tumor necrosis factor, interleukin 6 to regulate TH17 responses, and eventually all the other interleukins from 4 right through to 13 to activate eventually B cells to produce antibodies against the autoantigens such as myelin. So understanding MS through animal models has been extremely useful, and of course it has produced a lot of those drugs, as I mentioned before, going into clinical trials. So the autoantigens that can be present on the myelin include things such as, including integral myelin proteins such as myelin basic protein, proteolipid protein. It includes myelin oligodendrocyte glycoprotein, and various purified myelin constituents in their own right, where we actually get all the proteins and glycolipids together, which produce an immune response against them. And we utilize these proteins to produce the animal model in the disease, to identify the classical pathological hallmarks of MS in terms of demyelinating lesions and how the immune system enters into the central nervous system to produce those effects. We utilize those proteins or peptides of those proteins with an adjuvant to stimulate the immune response, added to that with pertussis toxin to not only expand the immune response, but also to allow for the breakdown of the blood-brain barrier, for cells to enter into the central nervous system effectively and produce their effector response. And of course, this generates a Th1 specific response, which produces the autoimmune encephalomyelitis model of multiple sclerosis. And we can delineate the inflammatory cell infiltrate very effectively, define the demyelinating events that occur subsequently for the immune cells once they enter into the central nervous system, and also the other bystander outcomes of axonal damage and eventual gliotic responses, which formulate the scarring of the lesions that appear on MRI in MS patients. So this is what we classically see in the animal model. After injection of one of the peptides that I described before, the MOG3555 peptide. It's a 20 amino acid sequence that produces a Th1 response in conjunction with complete Freud's adjuvant to expand that response. And over a time period of 30 days, we can see that it produces the classical clinical hallmarks of paralysis that is due to demyelination and axonal damage in this mouse model. So upon anywhere between 7 to 10 days, we get induction of disease and onset right through to the peak stage of disease that occurs around the 16 to 22 day period, and that's stabilized right through to chronicity, where we see a lot of those glial scar outcomes. Of course, it's an ascending paralysis, and it's a perfect model to test a lot of drugs to abrogate the immune response. So before I go to the next learning outcome, I wanted to give you a brief quiz at this stage in this small vignette of information I presented. So, which cells are likely to be dysregulated during the onset of multiple sclerosis? Oops. Promoting inflammatory infiltration within the central nervous system. Are they A, B lymphocytes in the spleen, B, T regulatory cells of the bone marrow, C, CD4 and MHC class 2 expressing T cells of the lymph node, or D, monocytic cells resident in the brain? So, of course, the answer is C, CD4 MHC class 2 expressing T cells of the lymph node, because it's these cells that expand the pro-inflammatory response occurring in MS. And as they exit out of the lymph node, they can expand that in the circulation and then enter into the brain to produce those pro-inflammatory responses that are seen. So in the next learning objective, we are now going to distinguish between disease-modifying therapies used to treat multiple sclerosis. So with regards to how we treat MS, we have, as I've said in the last couple of slides, we utilize these animal models. And the animal models have effectively shown us, just to recap, that we're capable of blocking the Th1 mediated response, and we can do that effectively either through injection of a cytokine like beta-ferron, interferon beta, or in fact stop by blocking using a monoclonal antibody the cells from transendothelial cell migrating into the central nervous system, such as tisabrin. This, of course, is effective in a lot of patients, but it's not effective in the vast majority of patients. So we can only get anywhere between 30 or 40% effectiveness in a lot of patients. But also, the disease is a continuum of disease, and once effective in terms of damage that occurs to the central nervous system, what we can classically see are those secondary outcomes where degeneration has occurred in myelin-forming cells and also in neurons specifically. And we now are starting to try to personalize medications for MS patients. A lot of it at the moment is a trial and error, trying to see which one is of greater effect in the vast majority of patients. But we also need more effective therapies in limiting the degenerative component of MS. Is when patients progress with a disease, they have substantial amount of neurodegeneration affected in their brains and spinal cords. And this is just a list of a lot of the drugs that have been listed for treating MS patients. And I discussed with you before about the beta-ferrons, where we're able to limit the capacity of the pro-inflammatory cytokines from affecting the disease, and so limit the Th1 mediated response from that perspective. We're also able to block the egress of the immune cells from the secondary lymphoid follicles, such as fingolimod, that is the modulator of the S1P, so sphingosine 1 phosphate receptor. And there are various other drugs, as I mentioned, natalizumab before. Don't worry about the others because a lot of these are given to certain patients to limit T-cell mediated effects, but haven't been as effective as what we'd like in a lot of the patients. So again, so this is just a brief list of those. One new drug that has been on the market for the last few years and is now on the PBS schedule is ocrelizumab, or Ocrevus. This is a really effective drug that's been utilized for progressive MS, but also in the vast majority of cases relapsing remitting MS, is to limit the B cell activity. So B cells being able to not only present antigen themselves, but also produce auto-antibodies in the process. Specifically, we've utilized a monoclonal antibody against a class of B cells, the pre-B cells, prior to plasma cells, not the plasma cells themselves, in maturing by antagonizing using a monoclonal antibody against CD20. And so we're able to limit the B cell response in the periphery and to a certain degree, those B cells that are entering into the central nervous system to produce those auto-antibodies, which can be quite damaging and harmful in patients. Of course, there are experimental therapies which have been devised, and in my laboratory, we're actually utilising a lot of these, but are still in either clinical trials or still very much experimental in preclinical models. And they include things like hematopoietic stem cell transplantation, which is currently being investigated in various hospitals such as the Alfred and also St. Vincent's Hospital and various other hospitals around the nation. These are relatively small trials, but they have undergone multi-centre evaluation. One of these major trials occurred in Canada, which showed substantial effectiveness of rebooting the immune system by transplanting their autologous hematopoietic stem cells of patients. So that is to say, you produce, you give a chemotherapeutic after taking out stem cells of those patients from the bone marrow. You give them a chemotherapeutic to limit the autoimmune cells and ablate the immune cells, the autoimmune potential memory cells in the hematopoietic stem cell pool, and then reinfuse the patient with their autologous stem cells to reconstitute their immune cell profiles. And it has had some effect, particularly in the relapsing remitting form and the aggressive forms of that disease. But it's usually used in conditions where the patients have been quite refractory to all of these other types of medications that I've presented to you just before and shown up here. So other potential stem cell approaches include mesenchymal stem cells, which have an immunomodulatory role, endogenous stem cells, where there's been one clinical trial that's been ongoing in the US, where they've injected endogenous stem cells, as well as embryonic stem cells into patients. And any experimental approaches such as inducible pluripotent stem cells. And the uniqueness of this is that it uses patients' own stem cells or cells that have the HLA match with that patient, so their own cells, to rederive stem cells that may produce a therapeutic outcome in patients when they're re-injected into the central nervous system, and that they can differentiate specifically into oligodendrocytes, for instance, that might be able to remyelinate the denuded axons following demyelination. So they're all experimental at this stage, and the efficacy of those are still being developed. So let's define lecture outcome 3, where we're here to analyse and evaluate the clinical and pathological presentation of multiple sclerosis. And this is quite a complex area, and I'll try and make it as simple as possible because MS neuropathology is quite involved. But we'll be discussing just the basic aspects of neuropathology in MS. So the basic aspect is the demyelination, quite clearly, in the scar formation that is evident on MRI. But in post-mortem analysis, what we can classically see is this graying of the white matter area. So in the middle here is all the white matter, which is the myelinated fibers. On the outside is the cortex, where all the neuronal bodies are in the brain. And what you can classically see here is that there is all this grayed area, which is the scar of the demyelinated lesion in the MS brain. And you can see that it's actually surrounding the ventricular space in this patient. If we do a horizontal section of this patient, you can see that the scar extends right throughout this whole area of white matter. And this is quite a profound, enlarged lesion in the MS brain. So, but there are multiple lesions like that right throughout the brain. And this is just a topography that demonstrates where we can find these lesions. I don't particularly want you to recall this. It's just to define how we can see that there are multiple sites, and that's why we define it as multiple sclerosis. Not only inside the brain, specifically in the white matter, we see all these scar formation, demyelinated lesions, as well as around the ventricles, but also in the brainstem. We can see it as transverse myelitis in the spinal cord. They can occur in the cerebellum, and they can occur in the optic nerves. So wherever there's white matter in the central nervous system, we can get demyelination affected. And of course, we can see variable signs of clinical manifestations in patients if they have demyelination in any one of these or multiple sites of these anatomical regions. For example, a demyelination occurring in the optic nerve, you might get blurring of sight, you might get diplopia, that means double vision, you might get black spots appearing in your visual field, or your acuity is off entirely. For example, if you have demyelination in the brainstem, you might get drooping of an eyelid or facial muscle, for instance. You might get other complex sensory symptoms, which a lot of patients present with if fibers of the somatosensory cortex in the brain are affected, or in the thalamus, or even in the spinal cord themselves. So any anatomical site where demyelination occurs obviously will present with their own variable neurological symptoms, and they're quite vast. But what is the cellular mechanism that's driving this? Well, of course, as I mentioned in the outset, yes, it's the T cell expansion of the immune activity, so expanded immune response. But once inside the central nervous system, the endogenous cells that produce the incredible amount of damage are the phagocytic cells. So the macrophages that end up from monocytes coming into the central nervous system to actively strip away that myelin and producing substantial amounts of extracellular debris, which is quite inhibitory to any repair mechanisms affected by the central nervous system, are the cells that are responsible. And you can see that these cells here are heavily laden with all this myelin debris as they're phagocytosing or stripping away, eating up all the myelin and engulfing it and entering into the cytoplasm. And so this blue stain specifically is staining for myelin because it has an affinity for fat. It's called the Luxor-Fast blue stain. But in the process of stripping away the myelin, we also get substantial amount of axonal damage. So we have neuroinflammation from once, which is propagated, and macrophages stay in the central nervous system for a long time, so beyond a month or so at any one time. And there's propagation of that neuroinflammatory response as a consequence. And also, eventually, as they strip away the myelin, they cause substantial amount of axonal damage by not only degenerating the axonal membrane, but also all the cytoskeletal structure inside the axon itself. So the axon dies back. It actually retracts back and degenerates. And of course, you lose connectivity. When an axon dies off, you lose connection from one neuron to the next. And this is a classical hallmark of that lesion. So we see a venule here, a central venule, and we have an immune profile around that venule because they've transendothelial cell migrated inside the central nervous system, and the immune cells themselves have produced substantial amount of this stripping away of myelin. So all this blue staining here is all intact myelin, but of course what you can see is this massive zone of right around the perimeter around the vessel here, where the immune cells have emanated from and are actively stripping the myelin away, shown here under high magnification from those macrophages. And of course, how do this occur in terms of demyelination, subsequent to demyelination for axonal damage? And this is classically shown in this in this video. It was supposed to be, but this is not working. I'll discuss that a little bit later before, but obviously axon damage occurs more effectively as a consequence of demyelination. You can see, obviously, the oligodendrocyte is the major cell of the central nervous system that myelinates axons. They can myelinate up to 40 axons. So one oligodendrocyte can myelinate up to 40 or so axons, and they produce extensive membranes around the axon to allow for proper signal propagation from the neuronal cell body right through into the axon for communication into the postsynaptic neuron that they interact with. And of course, once we lose this ensheathing area, this is where the axon then becomes susceptible for degeneration. And this is what's shown right here, where we see that myelin is becoming stripped away slowly, and we can see that the membrane itself is swollen, and also the axon as well, where the interaction between the myelin sheath and the axon itself is dysregulated. So in other words, the axon is also swelling, and eventually as the axon swells up, there's a substantial amount of signaling and mitochondrial dysfunction that occurs, and you see this mass amount of axonal swelling that occurs, and eventually this axon will be cut, will be transected, and so that we'll lose appropriate connection between one neuron and the next. And this is where we start getting all the neurological outcomes of those patients. Of course, there is remyelination that does occur spontaneously in MS lesions. They're very few and far between, but we do see it in MS patients. So wherever there's been myelin that's been stripped away, as shown in this panel here, where there's a lack of these proper integral myelin sheaths around axons, we do see some repair enhanced, but this doesn't last. So these, what we define as hypermyelinated shadow plaques, also end up degenerating over time with MS in MS patients. But this is because we see a slow expanding lesion after the first onset of symptoms of MS. And this is because we have innate neuroinflammatory processes affected as the patient lives with MS for many, many years. And what that means is that these activated macrophages and microglia are actively continuing to strip away myelin and also affect axonal damage. So substantial degeneration, and it ends up with this scarred lesion, which is low in cell density. So not only do we lose oligodendrocytes and myelin, but they're replaced with a glial scar, which is an astrocytic scar that produces extracellular matrix that is impenetrable for repair mechanisms to be affected. And as a consequence of that, because we have substantial loss of cells and cell membranes, which includes myelin, we end up getting these neurological clear pictures of an MS lesion, where over time, beyond 8 to 15 years, living with the condition, we see the reminiscent signs of atrophy of the brain. So the brain has actually shrunken down in this progressive form of the disease. The ventricles have swollen, so you get this compensatory hydrocephalus, and it's quite profound here as the patient has aged incredibly, and disability in those patients then are clearly affected. And we've lost a whole heap of axons, which is shown in this picture down here. And so this neurological progression continues on over time. So the classical patient begins with a relapsing remitting form, but ends up in what's defined as the secondary progressive multiple sclerosis phenotype. There are other forms of the disease where there's continued progression of the disease, continued decline in neurological status, but we won't define this in this lecture. In this lecture, we wanted to define the relapsing remitting form of the disease, where we get an onset due to a dysregulated immune system that affects the demyelination, and over time, the burden of disease occurs with substantial damage to the neural architecture in the brain and the spinal cord and the optic nerve. And so the patient inevitably, over many, many years, escalates in their disability status, where the clinicians use what's known as an expanded disability score system, and shows that there is no disability at the start at a score of 1, and minimal disability at 2, but as the disease progresses over time, the patient may be walking with a cane, or eventually requiring a wheelchair, a motorized wheelchair, or inevitably end up dying as a consequence of the disease. But they generally die of other disease mechanisms, particularly when they lose their coughing reflex, they might contract pneumonia. So here's a brief quiz for you. Quick question: how does the innate immune system contribute to the progression of the pathological outcomes in a person living with multiple sclerosis? Is it A, by enhancing axonal damage through Th1 cell-dependent mechanisms? Is it B, by enhancing chronic demyelination, axonal damage, and reactive glial activation? Is it C, by promoting acute inflammation through CD4 and MHC class II expressing T cells of the lymph node? Is it D, by activating peripheral monocyte infiltration to promote hyperactive demyelination? Well, the answer quite clearly is B, by enhancing chronic demyelination, axonal damage, and reactive glial scar activation, which we just talked about. So the innate immune cells, macrophages, and microglia promote those smoldering lesions and chronic demyelinating lesions over time, and the patient progresses with a disease. So what future therapeutic targets are we investigating then to limit the burden of disease? So I introduced you to some of these before, but remyelination is the great white hope for MS patients. Can we intervene therapeutically when we do see demyelinating lesions to repair that demyelination before we lose the axons? And what if we can do that along with have the immune cell, the autoimmune cells, I beg your pardon, in check, then we can have effective treatments for MS patients. So how can we remyelinate? We remyelinate by introducing endogenous cells that we can differentiate outside the body and then transplant into the patient. But the other mechanism of disease, which is a real problem here, is the glial scar formation, because we need to stop the sclerosing of those lesions to allow for any endogenous or any transplanted stem cells or precursor cells to affect a remyelination event to occur. And of course, as I mentioned earlier, these glial scars are quite inhibitory to any repair mechanisms occurring in the brain. Can we regrow axons? This is the major problem that we would see in a progressive patient. And in fact, by changing that extracellular milieu, which is degenerative in nature, maybe we can have some sort of effective regrowth of axons, because the neuronal cell bodies haven't died, but it's the distal ends of the axons that are degenerative. And so by stopping any signaling cascades occurring in the distal ends of the axons, maybe we can enhance their repair mechanisms to regrow through areas. So what have you learned in this lecture? We've outlined how dysregulation of the immune system may manifest inflammatory demyelination in the brain, optic nerve, and spinal cord. These are the anatomical sites that produce these clinical outcomes in patients. We have distinguished between disease-modifying therapies used to treat multiple sclerosis, and we've also analysed and evaluated the clinical and pathological presentation of multiple sclerosis. And I just wanted to leave this as an advertisement for my laboratory that's doing amazing work in trying to enhance the repair mechanisms in the central nervous system after inflammatory demyelination has occurred. And I thank you for your attention. As I said, my name's Stephen Petratos. I wanted to give you that overview of the immunopathogenesis of MS, and I wanted to finally describe who did you, or ask you, who did you believe Nancy was? Which one of the patients? Was it the young nurse that was talking to the older patient in a wheelchair? Or was it the older patient in the wheelchair talking to the nurse? Well, surprisingly enough, it was a nurse with a young onset of disease, so approximately 30 odd years of age. Nancy presented clinically with MS. She's Caucasian background, and she didn't present with the progressive forms of the disease. She progressed with a relapsing remitting form and had been medically managed to deal with her condition with some obviously residual symptoms ongoing over time. So Nancy has got some hope now because we have got some fantastic drugs to deal with her MS, but also hopefully in the future we'll have some better therapies to treat the neurodegenerative component of MS. Thank you for your attention, and hopefully I can answer any questions that you have through email or on Moodle.
Bueno, y esta fue la plática del doctor Steven Petratos, que nos habló sobre todos los avances científicos que hay de las enfermedades autoinmunes. Recuerden que si tienen alguna pregunta, la pueden enviar por medio del chat o en el grupo, y nosotros se la haremos llegar al doctor Steven Petratos. Igualmente será respondida en el grupo. A continuación, no te pierdas la plática de la licenciada Verónica González, caso de éxito de cáncer de mama por 2 ocasiones. La licenciada Fabiola Reyes nos estará acompañando para guiar la entrevista. Nos vemos en un ratito.